For some cancers, immune checkpoint inhibitors are, well, they're practically a miracle. You give the patient the drug, and it basically strips away the tumor's invisibility cloak.
Right.
And then the immune system just goes in and wipes out the cancer.
Yeah, that's the ideal scenario. But if you look at colon cancer, these same miracle drugs almost always hit a brick wall.
Right. They fail completely in the vast majority of patients. So today we're doing a deep dive into this incredibly fascinating new study from the journal Immunity. It was published in 2026 by Lobel and colleagues, and it suggests that the key to unlocking that brick wall might not be, another billion-dollar drug.
No, it might just be the specific amino acids on your dinner plate.
Which is wild. So welcome, everyone. I'm so glad you're here with us today for this deep dive.
It's a profound shift in how we think about oncology, really.
Okay.
Today, we're tracing a highly specific biological circuit. We're going to explore how a surprisingly simple dietary tweak can completely reprogram your gut microbiome.
And then that sends a chemical flare into the immune system, right?
Yeah, exactly. It trains a microscopic army to shrink colon cancer. And the best part is, we're looking at this through the lens of reverse translation.
Reverse translation, meaning we aren't just starting with some cells in a petri dish and hoping it works in humans decades from now.
Precisely. We start with a mystery hidden in massive amounts of human clinical data. Then, we dive deep into highly controlled laboratory mouse models to find the exact biological mechanism, literally mapping it cell by cell.
And finally bringing it all the way back to validate it in human cancer treatments. I love that approach.
It's the gold standard.
Okay, I am fully ready for this. We are going to keep all the juicy, rigorous science, but I want to make sure we focus on how and why these mechanisms work, skipping the dense acronyms that usually just bog these papers down.
Sounds like a plan.
Awesome. So before we can figure out how to fight colon cancer, we have to understand what successful cancer fighters actually have in common, right? So let's start with the big picture and those human clues.
Well, the researchers began with a massive meta-analysis. They really wanted to know why a very small fraction of people respond beautifully to these immunotherapies, while most just don't.
Right, because if you can figure out what's different about the responders, you might have a totally new target.
Exactly. So they analyzed 17 different immune checkpoint inhibitor studies. This covered melanoma and lung cancer, over 1,400 patients in total.
Wow, that's a lot of data.
It is. They compared the gut microbiomes of people who responded well to the therapy versus those who didn't. And on top of that, they pulled metagenomic data from 12 separate colon cancer cohorts.
So they're looking at the gut bacteria of over 900 colon cancer patients against over 900 healthy controls. Okay, let's unpack this. What exactly were they looking for in all that data?
Right, so let's set the baseline. For someone who might just be catching up on the immunology, we should probably define how exactly an immune checkpoint inhibitor in ICI actually works.
Yeah, that would be helpful.
So, your immune system has built-in brakes. These are checkpoints that stop your immune cells from attacking your own healthy tissue.
Basically preventing autoimmune diseases, right?
Exactly. But tumors are incredibly smart. They hijack these brakes, they wear them like a molecular invisibility cloak, essentially telling the immune system, hey, nothing to see here, move along.
And the ICI is the drug that blocks those brakes.
Right. It rips off the invisibility cloak so your immune killer cells can finally see the tumor and attack.
But earlier, we established that ICIs hit a brick wall when it comes to colon cancer. Why is the colon such a blind spot for these drugs?
There are two main reasons. First, the colon itself is naturally a very tolerogenic environment.
Tolerogenic, meaning it tolerates things.
Exactly. Think about it. Your colon has to tolerate massive amounts of foreign food and trillions of bacteria every single day without constantly triggering massive destructive inflammation. It is basically programmed to be overly tolerant.
Oh, that makes perfect sense. If your colon's immune system was on a hair trigger, you'd be in agonizing pain every time you ate an apple.
Precisely. And the second reason is that the vast majority of colon cancers are what we call MMR proficient, or mismatch repair proficient.
But what does that mean for the cancer?
It means the cancer cells are actually still pretty good at repairing their own DNA. Because they repair their DNA, they don't have a high mutation rate.
I see.
And to the immune system, a cell with very few mutations doesn't look very foreign. It just looks like normal colon tissue.
So even if you took the brakes off with an ICI, the immune system looks around and doesn't see anything worth attacking.
You've got it. That's the brick wall.
Man, so colon cancer is hiding in an environment that is biologically wired to ignore intruders, and the cancer itself doesn't look strange enough to trigger an alarm. That is incredibly stubborn.
It really is. Which brings us back to the human data. In all those thousands of patients, they wanted to know what the healthy people and the rare ICI responders actually had in common.
Right. And what did they find?
They shared a highly specific feature in their gut microbiomes. Their gut bacteria had incredibly high levels of sulfur amino acid metabolism.
Sulfur amino acids. We're talking about SAW here.
Yes. Specifically, these microbiomes were highly active in processing two specific amino acids, methionine and cysteine.
Wait, hold on. Sulfur is everywhere in a standard diet. Methionine and cysteine are in, like, eggs, meat, dairy.
Yeah.
If it's just about eating more sulfur amino acids, why wouldn't a standard high-protein diet show the same protective effect across the board? I mean, it can't just be a magic switch, right?
That is the critical distinction, actually. It is definitely not a magic switch. And just eating a steak doesn't cure cancer. It's about precision ecology.
Precision ecology, I like that.
It's about strategically altering the gut environment to prime the immune system. A standard high-protein diet comes with a massive mix of other nutrients, fats, and competing amino acids that feed 1,000 different types of bacteria.
Ah, so it gets drowned out.
Exactly. What the researchers suspected was that a very specific isolated ratio of these sulfur amino acids might be feeding a hidden ecosystem that alters immune function.
Okay, so just to give you, the listener, a quick recap of where we are so far, let's do some bullet points. Number 1, ICIs fail in most colon cancers because the colon is tolerant and the tumors are basically invisible.
Right.
Number 2, the rare patients who actually do respond to these drugs all share a microbiome that is heavily processing sulfur amino acids, which leads to a question for you, the listener. Have you ever considered that the specific amino acids in your meals might be feeding a hidden ecosystem that basically controls your immune system?
It's a fascinating concept, but seeing a correlation in human data is just a clue. To prove that a specific diet actually restricts cancer, we need rigorous evidence grading.
Right, because since we saw the sulfur clue in humans, we need to test it rigorously. You can't just feed human patients experimental ratios of sulfur without knowing the exact mechanism, right, or the safety profile for that matter.
Precisely. That brings us to the controlled mouse models. The researchers designed two specific isocaloric diets, meaning they had the exact same number of calories.
Got it. To rule out just weight loss or calorie restriction as the cause.
Exactly.
Yeah.
One was a low sulfur amino acid diet, and the other was a high sulfur amino acid diet. And the safety parameter here is vital.
Because too much sulfur is toxic.
Yes, if humans consume way too much methionine, it can lead to a toxic condition called hyperhomocystinemia, which damages blood vessels.
Oh, wow. Okay.
The high diet in this study was formulated to be perfectly safe, well below that toxic threshold. And the low diet wasn't starvation either. They didn't completely remove methionine because that would just crush the immune system anyway.
So these were physiologically realistic diets, things a human could actually do safely. How did they test it?
They tested it across two different levels of evidence. The first was a baseline model called the MC38 model. They essentially injected colon cancer cells just under the skin of the mice.
Under the skin. Why not in the colon?
It's a widely used heterotopic model. It's relatively easy for the immune system to recognize because the cancer is MMR deficient, and it isn't growing in the colon where that extreme immune tolerance exists.
Okay, so it's sort of a proof of concept. What happened on the high-Saa diet?
The mice on the high-Saa diet saw a 50% reduction in tumor volume and a 30% reduction in tumor weight compared to the low diet.
50%. That's a massive reduction just from tweaking amino acids. But you mentioned that was the easy baseline model. What about the stubborn, invisible colon cancers we talked about earlier?
Right. To test that, they brought in a genetically engineered boss-level model called the CAPC model.
Why do we need a second model? What makes this one the boss level?
They use specialized genetic tools to delete a specific tumor-suppressing gene exclusively within the colon tissue of the mice. This perfectly mimics how about 85% of human colon cancers actually develop naturally.
Wow, so it's a completely accurate simulation.
Yes. It creates a tumor that is MMR proficient, meaning it has low mutations and looks completely normal to the immune system, and it grows right inside that highly tolerant colon environment. It is the ultimate treatment-resistant test.
The absolute brick wall. So how did the high-Saa diet perform against the boss-level cancer?
The results were staggering. The high-Saa diet caused a massive fourfold decrease in the occurrence of advanced adenocarcinomas.
A fourfold decrease?
Yeah. And the tumors that did manage to form were half the weight and size of those in the low diet group.
Let's really pause on that for a recap. Let's look at the bullets. Bullet 1, a highly specific, safe, high-Saa diet shrinks tumors. Bullet 2, it works in both the easy MC38 model and the incredibly stubborn CAPC colon cancer model.
Which is huge.
It is. So think about this for a second. If you were a scientist designing a clinical trial, wouldn't you want a dietary intervention that works even on the most treatment-resistant tumors?
Might. There is a glaring missing link here.
What's that?
The sulfur amino acids are going into the stomach and the gut. They aren't like circulating in the bloodstream, hunting down cancer cells with little swords, right?
Yeah, they aren't attacking the tumor directly at all. They have to go through a middleman. Here's where it gets really interesting. What actually changed in the gut environment when they introduced this high sulfur feed?
Well, the researchers looked closely at the physical lining of the colon, specifically the mucus layer.
Okay.
They found that the high-Saa diet physically thickened the colonic mucus. And what's fascinating is how it did it. It didn't alter mucin gene expression, like it didn't activate the NUCA2 gene to magically command cells to manufacture more mucin proteins.
So it wasn't a genetic change?
No, the abundant sulfur just provided the physical building blocks to reinforce and beef up the mucus structure itself.
So the sulfur literally acts like a biological fertilizer for the gut lining. You just get a thicker, more robust mucus barrier. But how does a thicker wall of mucus stop a tumor?
It creates a highly specific habitat. If we use an image memory trick here, imagine that thick mucus as a lush, dense canopy in a rainforest.
Okay, I'm picturing it.
By thickening it, the diet created the absolute perfect ecosystem for one very specific microbial middleman to bloom, a bacterium called Mucispirillum schaedleri.
Mucispirillum schaedleri. That's quite a name. What is its role in all this?
It's a gram-negative anaerobic bacterium that naturally lives within the mucus of the colon. When the high-Saa diet provided that lush mucus canopy, this specific bacteria multiplied rapidly.
It just bloomed.
Exactly. And if we look back at human biopsies for a moment, the abundance of this exact bacteria negatively correlates with colon cancer.
Wait, meaning what exactly?
In normal, healthy human colon tissue, it is highly abundant. But in patients with advanced colon adenocarcinomas, it is completely absent.
Oh, wow. So when the cancer takes over, this bacteria is just wiped out, but the high sulfur diet brings it back in full force.
Right.
Okay, time for another quick bullet recap. First bullet, the high-Saa diet physically thickens the colon's mucus layer. Second bullet, that thicker mucus allows this highly protective bacterium, M. schaedleri, to rapidly multiply. So listener, what stands out to you about the fact that a simple dietary amino acid can act like fertilizer for just one specific cancer-fighting bacteria? It's not just taking a generic over-the-counter probiotic. You are feeding the soil to grow a specific microscopic guardian.
It's a great analogy. But now we have a blooming population of bacteria sitting in the gut mucus, while the cancer is growing deep in the colon tissue.
Right. The immune system operates out of the lymph nodes. How does a bacterium just hanging out in the mucus tell the immune system to launch a targeted attack on a tumor?
This brings us to the absolute core mechanism of the study. We can break this down into a highly orchestrated relay race, a 5-step biological circuit.
This is the dense part, so let's do a line-by-line breakdown. Walk me through the handoffs. How does the signal start?
Step 1. The ballooning M. schaedleri bacteria secretes specific organic molecules into its environment. The researchers analyzed these and found they are heat labile.
Heat labile, meaning they break down under high heat.
Yes, which tells us they are complex, delicate organic structures.
Okay, so the bacteria is pumping out a chemical signal into the gut. Step 2.
Step 2. Those secreted molecules diffuse across the barrier and reach the lymph nodes that drain the tumor area. And waiting in those lymph nodes are natural killer T cells, or NKT cells.
NKT cells.
Yes. When the molecules reach them, the NKT cells become highly activated.
I know standard T cells are part of the adaptive immune system, the learned response. What makes a natural killer T cell different?
NKT cells are fascinating because they bridge the gap between your innate, rapid response immune system and your adaptive, highly targeted immune system.
Ah, I see.
They're like special forces scouts. They don't need days to learn what an enemy looks like. They can rapidly spot specific molecular patterns and immediately sound the alarm.
Okay, so the bacterial molecules cross over and activate the special forces scouts. That's step 2. What is step 3? What do the scouts do once they're awake?
Once activated, the NKT cells secrete a massive amount of a highly specific chemokine called XCL1.
A chemokine. So that's basically a chemical messenger that creates a trail. Let's use another analogy here. I like to think of XCL1 as a chemical flare gun. The scouts have verified the signal from the gut bacteria, and they fire this brilliant XCL1 flare straight up into the dark to call in the heavy artillery.
That's a perfect analogy. And that leads to step 4. That XCL1 flare specifically recruits a very rare type of cell called a CDC1, a type 1 conventional dendritic cell.
Okay, so the flare calls in the CDC1s.
Yes. The flare guides them directly into the tumor-draining lymph nodes. You can think of the CDC1s as the master generals of the immune system.
Master generals. What makes them the generals?
They excel at a complex process called cross-presentation. They are uniquely capable of picking up pieces of dead tumor cells, processing them, and presenting them directly to the immune system's killer cells.
So they are essentially handing out high-resolution mugshots of the cancer to the assassins.
Exactly. And that brings us to the final step, step 5, the assassins.
The CD8 plus T cells.
Right. Once the generals activate them with those mugshots, the CD8 plus T cells infiltrate the tumor in huge numbers. And because of this specific signaling chain, they arrive heavily armed.
Heavily armed with what?
They express incredibly high levels of granzyme B, which is the toxic payload they inject into cancer cells to destroy them.
Oh, wow.
And crucially, they also arrive fresh. They showed significantly fewer exhaustion markers.
Exhaustion markers.
Yeah, proteins like PD-1 and LG3 that usually force T cells to shut down prematurely.
So they are armed to the teeth and they aren't tired. Okay, let's trace this incredible 5-step chain of events for our recap. Bullet 1, the bacteria secretes a delicate molecule. Bullet 2, that molecule activates the NKT cell scouts. Bullet 3, the scouts fire the XCL1 chemical flare. Bullet 4, the flare recruits the CDC1 master generals. And bullet 5, the generals unleash the CD8 plus T cell assassins directly into the tumor.
That is the complete circuit.
It is an elegant mechanism. But immunology is famously messy. I have to push back here. So what does this all mean? How do we know this exact chain of events is real and mandatory, and not just a bunch of random coincidences happening at the same time?
That is a great question. That is the gold standard of immunological research, and the researchers proved it by conducting exhaustive knockout studies. They systematically broke every single link in that chain to see if the diet still worked.
How exactly do you break the chain in a living animal?
Let's look at the master generals, the CDC1s. The researchers used mice that were genetically engineered, specifically BATF3 knockout mice or ZBTB46 DTR mice, to completely lack the ability to produce CDC1 cells.
Okay, so no generals. What happened?
When they put these mice on the high-Saa diet, nothing happened. The cancer grew unimpeded. Without the generals, the diet was useless.
Wow, what if you take out the flare gun?
They did that too. They injected mice with neutralizing antibodies specifically designed to bind to XCL1.
So literally plucking the flare out of the sky before anyone could see it.
Exactly. The result, no generals arrived, no assassins were deployed, and the diet failed to stop the tumor.
And what if you remove the scouts at the very beginning?
They used a specific antibody, the NKT14 antibody, to completely deplete the NKT cells. Without the scouts, no flare was ever fired. The cancer grew. Every single link in this relay race is absolutely mandatory for the anti-tumor effect.
If one piece is missing, the whole circuit collapses.
Exactly.
That is just brilliant, rigorous science. So, listener, picture this microscopic relay race. Which specific link in that complex chain surprises you the most? Because for me, it really highlights how communication is just as critical as the killer cells themselves. You can have all the assassins in the world, but if the flare gun is never fired, they never get the mugshot.
It's so true. The immune system is entirely dependent on context and signaling. But curing mice is great. It's a monumental achievement, but it's not the ultimate goal.
Right. The listener listening right now cares about human health. We have to bring this reverse translation full circle. How does this beautiful molecular circuit apply to us?
What's fascinating here is that the researchers didn't stop with the animal models. They took conditioned medium, which is essentially the liquid containing all those secreted organic molecules for the M. schaedleri bacteria, and they applied it directly to human NKT cells.
Human cells isolated from peripheral blood.
Yes.
Did the human scouts recognize the bacterial signal? Did they fire the flare?
They did. The human NKT cells successfully stimulated massive production of the XCL1 flare, just like we saw in the mice.
Oh, wow.
The core mechanism is evolutionarily conserved.
That is a massive validation. But what about the generals? How do we know the human CDC1s respond the same way?
To figure that out, the researchers look at the genetic programming of the cells. They performed single-cell RNA sequencing, or cRNA-seq on the master generals from the mice.
Which lets you read the exact genetic blueprint of an activated cell.
Right. Exactly. They discovered that mice on the high-Saa diet had generals with a highly unique, intensely activated inflammatory gene signature. They called it the HSME signature.
HSME, what does that stand for?
It stands for high-Saa M. Schaedleri exposed.
Okay, so they isolated the exact genetic fingerprint of a master general that has been successfully recruited by the diet and the bacteria. What do you do with a fingerprint like that?
You look for it in humans. They took that specific HSME fingerprint and search for it in the TCGA database.
The Cancer Genome Atlas.
Yes, which holds the genetic data for thousands of real human cancer patients. They specifically filtered for human colon cancer patients. And they found that patients whose tumors naturally express this highly activated signature live significantly longer.
Wait, let that sink in. A genetic signature discovered in a mouse eating a highly specific amino acid diet perfectly predicted survival rates in human colon cancer patients.
It's incredible. And it gets even better when we look at active clinical treatments. The researchers examined data from a clinical trial involving human melanoma patients.
Yes.
These were patients who had completely failed standard immune checkpoint inhibitor therapy. Their cancers were resistant. In the trial, these patients were given fecal microbiota transplants, or FMT.
FMT. Basically, a total microbiome transplant from a healthy donor to try and overcome the drug resistance.
Yeah.
Did the transplant work?
For a subset of patients, yes.
Yeah.
And when the researchers analyzed the genetic data from the patients who actually responded to the treatment, they saw massive increases in both the NKT cell signature and that specific HSME master general signature post-FMT.
Wait, really?
Yeah. The exact biological circuit we just mapped out was actively turned on in the human patients who survived.
So just to pull these threads together for our final recap. Bullet 1, the chemical circuit works on human immune cells in a dish. Bullet 2, the specific HSME gene signature predicts significantly longer survival in human colon cancer. Bullet 3, in real-world clinical trials, patients who respond to FMT show this exact same immune activation.
It's a perfect loop.
So, listener, if a mouse's dietary immune signature can predict human cancer survival, how might this fundamentally change the way we view hospital food and personalized nutrition?
If we connect this to the bigger picture, it fundamentally reframes how we understand medical nutrition. Nutrition isn't just about giving a patient enough basic calories to keep their weight up during chemo.
Right.
Nutrition is a highly complex language of chemical signals that directly programs our microbiome, which in turn programs our immune system's ability to fight disease.
This whole journey is just a masterclass in reverse translation. We started with a murky mystery in human clinical data. Why do some people respond to immunotherapies while others hit a brick wall?
And we found the clue: sulfur amino acid metabolism.
Then we dove into the mouse models and meticulously mapped the exact mechanism. Diet thickens the mucus blooms the bacteria activates the NKT scouts fire the XCL1 flare. The flare recruits the CDC1 generals, and the generals unleash the CD8 plus T cell assassins.
And finally, bring it back to validate in human cancer patients.
When you think about hospital food, you usually just think about basic sustenance. But this completely flips that logic on its head. You aren't just feeding a patient, you are actively programming their immune system.
We are rapidly moving away from treating a tumor in isolation. The tumor, the immune system, the gut bacteria, and the diet are all one interconnected web.
And I want to end on a provocative thought for you to mull over today. If specific amino acids can act as targeted fertilizer for specific bacteria, which then turn on specific immune pathways, could the future of oncology involve designer diets?
That is the big question.
Imagine perfectly curated meals and personalized probiotic pills mapped seamlessly to your unique gut ecosystem, unlocking the power of immunotherapies for cancers we previously thought were totally untreatable.
That is exactly the frontier we are looking at.
A completely new way to arm the immune system, starting from the inside out. Thank you for taking this deep dive with us. Until next time.